Good afternoon and welcome to the Jones Seminar. We have a special event, tag team match, I think they call it. And what we have done is we've asked the students who gave presentations in the winter term and whose talks were rated most highly by their peers to expand those talks to inform the community somewhat of the research that's been going on here at the Thayer School. So we're going to have two talks. There will be time for a couple of brief questions between the talks, and then the speakers will stay around afterwards if there are additional questions. So the first talk that we have is by Alex Bijamov. He's a third-year Ph.D. student here in electromagnetic sensing group, and he received his BS in physics from Tbilisi State University in the Republic of Georgia in 2006. In 2002, he was awarded bronze and gold medals at the 3rd Asian and 33 International Physics Olympiads in Singapore and Indonesia, respectively. His research interests are electromagnetic sensing and computer simulations for biomedical research, and including DNA sequencing, which is what he's going to talk about today. So please welcome Alex.
Can you hear me? Now? Hello? Hello? DNA? DNA? Yeah. Hello, everybody. Thank you, Professor Gibson, for introduction. And thank you all for coming here and for good feedback during Research in Progress workshop. My talk today will be about our way towards $1,000 human genome sequencing using force spectroscopy. And it's organized in the way that I will first talk about DNA sequencing in general, then introduce our sequencing method using force spectroscopy, and finally tell you about the challenges we're encountering and the computer simulations we're doing to do the sequencing. So first of all, what is DNA? DNA is a long molecule which contains genetic instructions used in development and functioning of all living organisms. Physically, DNA consists of two strands, a sugar phosphate backbone, which warp around each other to create a double helix. And to each strand, there are 4 types of bases attached. They can be of types A, T, G, or C. And the specific binding between these bases requires that if we have base A on one strand, it binds only to base T on the opposing strand. And similarly, if we have base G on one strand, it binds only to base C on opposing strand. Now, the information in DNA is written along each strand, but since they're complementary, in a sense, if we know sequence on one strand, using this ATGC rule, we can reconstruct the sequence on another strand. And to sequence the DNA means to read out this sequence of letters in the entire DNA molecule. And for human genome, it contains 3 billion base pairs. So to sequence the genome of one person means to read out all these 3 billion base pairs. Now, why do we need this? Because once we have sequenced a lot of people and know their whole genomes, it will create an enormous playground for researchers to identify genetic differences in sickness and health, and open the doors to personalized medicine where the drugs can be designed and prescribed for a particular person for targeting particular disease in a particular gene. But to do this, we need to sequence a lot of people together and reach affordable and fast human genome sequencing. So let's see how it was evolving, how human genome sequencing was evolving. The first project to sequence one person's DNA molecule was proposed in 1995, and it was proposed to estimate to cost $3 billion to sequence this molecule from 1990s till 2005. But this cost of $3 billion included not only the sequencing process itself, but also all accompanying research in medical devices creation and study of legal and ethical issues of DNA sequencing. And by 2003, when the project was complete, it took 13 years to sequence it, and the total cost of pure sequencing process was $30 million. By 2008, the constant sequence refinement of technology caused the price to drop down to $2 million. The company called 454 Life Sciences sequenced one person's genome within 2 months for only $2 million. And you can see it's a huge huge reduction in price and increase in time, 13 years and 2 months. But it's still not enough. To truly benefit from paradigm of personal genomics, we need to come up with even cheaper and even faster genome readouts for whole human genome. And current goal is to achieve $1,000 human genome sequencing available within several hours using a tabletop setup. Now, what are the obstacles towards this? The process of DNA sequencing used up to now are the following main processes. So first of all, there is Sanger sequencing, which was used in the first human genome sequencing project. And it relies on stages of amplification, massive cloning of DNA molecules, and then fluorescent tagging of individual nucleotides, a huge number of individual nucleotides with fluorescent labels. And then it separates them with gel electrophoresis to create a readout. Now, these two processes of massive cloning and fluorescent tagging are very expensive and time-consuming. And some of the reductions in price and boost in time came from transition from such techniques towards single molecule label-free techniques. And one of them is pyrosequencing. Actually, it still requires a step of massive cloning of DNA fragments, but it eliminates the fluorescent tagging requirement by observing not the color of the tags, but in fact, the release of inorganic pyrophosphate during single nucleotide addition. And pyrosequencing was used by 454 Life Sciences to sequence the second human genome. Now, third technique currently under development is called nanopore sequencing. It is truly single molecule, label-free technique, and it's based on the physical differences between individual bases, A, T, G, C. The single strand of DNA molecule is pulled through a nanoscopic pore, and depending on which nucleotide is currently inside the pore, we observe, it's possible to observe different electric currents through the pore. And by observing it in time, one can say which base is currently being sequenced. But this method encounters huge noises and issues with pore stability, and it's very difficult to predict how many bases are currently inside the pore, and it's still being developed. The question is there any other physical property on which we can base our sequencing instrument? And turns out there is. There was huge research done into study of force extension responses of DNA molecules, basically to study the rigidity properties. And it turns out that single-stranded DNA and double-stranded DNA molecules have different rigidities. If we stretch them with certain force, single-stranded is longer than double-stranded. Of course, because double-stranded is more rigid. Now, if we have a composite molecule which contains a part of single-stranded DNA and double-stranded on the bottom, and we start adding one base pair into this boundary to slightly elongate the double-stranded part, we will see the total length difference, total shrinkage of this entire long molecule. And this length difference is sub-nanometer scale. Now, if it's possible to detect this sub-nanometer scale length difference, it would be possible to detect single nucleotide addition. And one could base sequencing technology on this. But before we go to actual process, let me tell you a couple of words about how the second DNA strand is being built around the first one. Many people ask questions about it. So assume we have this blue single-stranded DNA molecule as a template, and we want to construct a full double-stranded DNA molecule based on this template. And assume we already constructed a certain part of it up to here. Now, in chemistry, it's well known there is an enzyme called Dna Polymerase, which is basically a tool. It sits over here on the boundary between single and double-stranded DNA molecules, and it can attract a certain nucleotide from the surrounding medium and attach it here to the single-stranded template. And this nucleotide is actually the one which is supposed to be on the double-stranded part according to the binding rule, ATGC. Now, the problem with DNA polymerase is that no known DNA polymerase can initiate the synthesis of a second DNA strand. It can only continue the existing strand to build double-stranded DNA. So how do we start it? Turns out there is another enzyme called primase, whose purpose is to create a short primer. It binds on the starting part of the single-stranded DNA molecule and creates a primer which can bind to this point. And after that, the DNA polymerase can continue the process, building up the entire double-stranded DNA molecule from bottom up. Now, this is how our sequencing works. Assume we have a DNA molecule, single-stranded DNA molecule, attached to a solid substrate, and we're pulling it with this spherical bead, which is microscopic scale, by using magnetic tweezers. The bead has magnetic loading and fluorescent fluorophore. And assume we have already sequenced a certain part of the DNA molecule up to here, and here is our DNA polymerase. And now we want to know which base pair is here. Question mark. What do? We have only 4 letters in the alphabet, A, T, G, C. We take all A nucleotides, put them into the solution using microfluidic channels, and see what happens. Well, A nucleotide nothing happens. So A nucleotide is not next in sequence. And what do? We wash them out and try some other nucleotide, G, C, T. So if C nucleotide is complementary to the existing part of the single-stranded template, it will be attached. And during this attachment, since now we have slightly longer part of the double-stranded DNA molecule in the entire string, we will have a tiny sub-nanometer decrease in the total length of the DNA. And if we detect this decrease in DNA length, we would be able to say that, yes, single nucleotide addition occurred, and since we tried to put C-type nucleotide, therefore, the complementary one should be G, and therefore we sequenced the G nucleotide, and we move on further up to sequence the rest of the DNA molecule. So our DNA sequencing method rests on the ability to detect this small sub-nanometer scale length change of the long molecule. How do we do this? This is the experimental setup our colleagues use in Lehigh University. It basically contains the chamber, liquid chamber, which hosts 200 base pair long parts of a single DNA molecule arranged in a massively parallel format. And to each free end of the DNA molecules, we attach a microscopic magnetofluorescent bead, which is pulled using magnetic tweezers. And we create the total internal reflection evanescent field in the liquid chamber. And therefore, when the bead is close to the surface, it is in the regions with high electric field, and it absorbs and emits high intensity of light, which we detect by CCD camera. When the bead goes deeper into the chamber, therefore the DNA molecule becomes longer, we observe that the beads become fainter. And by looking at the intensity of the bead, we can tell how deep it is. Now, the problem is that we need to detect by observing only intensity of the beads. On this figure, you can see each point corresponds to a particular bead at a certain distance inside the chamber. We need to detect sub-nanometer scale changes in DNA lengths based on the intensity changes of these images. And for these purposes, we need to optimize both magnetic tweezer instrument to provide high pulling forces and be in the regime where the differences are highest, and also optimize the bead loading and other parameters of the setup to ensure that we have high sensitivity to detect these small changes. We do this optimization numerically using the method called method of auxiliary sources. It's basically a numerical tool which solves Maxwell's equations in electrodynamics subject to boundary conditions. And the difference from other boundary value methods is that it uses representation of fields in the domains of interests by representing fields created by auxiliary sources, but these sources are not located on the boundaries of the objects, but are shifted and are on the virtual surfaces inside and outside of the objects. Well, to cut long story short, it is a great method. It's very easy to implement, and it's very accurate to solve the electromagnetic scattering and absorption problems. Let's see what we can do with it. First of all, when our colleagues from Lehigh decided to make a pulling experiment, they purchased a mu metal core to create a magnetic tweezers instrument, put a coil around it, and they expected that when they run the current through the coil, the DNA molecules will stretch vertically up, and these beads will be pulled. But in fact, the pulling force was very small, and the beads were going all variety of directions, but not upward. And what's the problem? The problem was, that's the last formula for today, I promise. The problem was that the magnetic pulling force depends not on the field itself, but on field gradient. And over here, you can see if the metal core has a flat tip, you will have uniform magnetic field, which has 0 gradient, and the pulling force is small. On the other hand, on the edges of the core, where the curvature is high, the gradient is higher, and the beads are pulled towards the edges of the magnet. So we gave it a thought and decided to create a magnet with a single tip and study the geometry, how the geometry of the magnet, particularly the length, radius of the core, and the length and radius of the tip, affect the pulling forces. And here you can see the magnetic field distribution inside and outside of the magnet. Since it's high permeability magnetic core, most of the magnetic field is located inside the magnet. But every magnetic material is characterized by saturation. After some time, if you keep increasing the current inside the coil, you will not get further increase in magnetic field. For mu metal core, it happens at 0.6 Tesla magnetic fields. And the only way we can now increase the pulling forces on DNA molecules is to modify the geometry of the magnet to target the high magnetic pulling forces onto this region where the DNA molecules are located. And if we blow it up, you will see over here this small region where DNA molecules are. And you can see that indeed the pulling forces are pretty vertical in a small region here, away from magnet tip. But the problem is they're small. It's only 15 piconewtons in our case. To be in a good regime, pulling regime, to see the visible good changes in DNA lengths, we need to provide 60 piconewtons pulling force. So we studied what has an effect on the pulling force. Turned out the magnetic tip radius has the most effect on it. And you can see that when we reduce the magnetic tip radius curvature, the fields in the vicinity of the magnet increase. It's similar effect to electrostatics, so it's no surprise. And the second observation is that this fact depends on the distance from the tip. So if we are far away, further than 1 millimeter, it turns out that the magnet with curvature of 1 millimeter overpowers the magnets with small curvatures. So depending on the distance of our DNA samples from the tip of the magnet, we can change the magnet geometry to target the highest possible pulling force on that particular region. And the other problem is that the smaller is the curvature of the tip, the higher are the lateral pulling forces on the beads. We want the pulling forces to be mostly vertical, and we don't want to allow more than 5% in lateral pulling components. So we studied how the force uniformity depends on the radius of the tip and other parameters of the magnet, and decided to use magnets with radius of the tip of 1.5 millimeters, machine the magnet, and check its performance. And it matches pretty well with our numerically predicted calculations. And now this magnet is used in our prototype DNA sequencing device at Lehigh University. Now to the second part, imaging. How do we find the position of the bead inside the liquid chamber? Well, as I said, we create the total internal reflection is happening on substrate-water interface, and the evanescence field is created inside the chamber, which is absorbed by beads' magnetic and fluorescent components. Now, on the second stage, during fluorescence stage, the part of the energy which was absorbed in magnetite is dissipated into heat, and the part which was absorbed in fluorescence is emitted back. And after passing through water substrate, is collected by the objective and converted to our visible screen image. Now, what we need to do is we need to optimize the bead composition and the setup to ensure that we get detectable power, and that this power, the observable power of the bead, covers the entire range of DNA extensions. On this figure, on the video on the left, you can see the simulated image of the bead on the screen. You can see that when the pulling force on the DNA molecule increases, the bead goes deeper inside the water chamber, the DNA molecule becomes longer, and the image becomes less, the intensity of the image drops. And when our colleagues from Lehigh conducted an experiment, they were changing periodically the current through the electromagnet, thus increasing and decreasing the pulling forces. They observed that the image of the 2 micrometer diameter commercial bead attached to 150 base pair long single-stranded DNA is following the pattern of the current. Therefore, this result shows that we can repeatedly manipulate DNA molecules, and this is one of the big steps towards the successful sequencing of the DNA. But our task right now is to ensure that the intensity change corresponding to sub-nanometer scale length changes of DNA molecules can be detected. And for these purposes, we need to study the fundamental response of magnetofluorescent beads, micrometer-sized beads, on Z micro and nano scales. We use the method of auxiliary sources to study the behavior of fields in and out around the beads. On this figure, you can see the power which flows through the substrate vertically down and is collected by the objective transformed to the screen image. As you can see here, it's a log scale. When the bead goes up and down inside the chamber, the power changes as predicted. And we need to quantify this change. Also, I want to show you these figures. These are electric field distributions inside and around the beads. This shows the plane of incidence of electric laser beam, and this is perpendicular plane. You can see a couple of observations. On this figure, you can see that when bead goes deeper and deeper into the solution, the intensity of the magnetic electric fields inside the bead drops, and therefore the fluorescent power also drops, as expected. And also, there are certain places inside the bead close to its center where nearly no power is observed. 1,000 times less power is absorbed inside than it is absorbed in the outer shells of the bead. And on this picture, you can see that there is a certain amount of power which is radiated in upward direction. Potentially, it can be collected and analyzed, but in our simulation, in our experiment, we are collecting the power which is going down, collected by the lens. Now, how do we quantify this power? Well, on this figure, we can see the dependence of power on bead depth, and the left figures show the powers which are absorbed by the bead and the power scattered through glass on absorption stage, and the right plot shows the powers emitted from the bead and transmitted through glass, actually absorbable power at fluorescent stage. In our region, from 0 to 500 nanometers extensions, all these powers are exponentials, and we can calculate decay lengths. It's the distance over which the intensity, observable intensity of the bead decreases by one exponent. Now, if we don't have any bead, if we have only a luminescent field created at glass-water interface, we would have the decay length of 97 nanometers. But the presence of the bead perturbs this field, and decay length may change. What we observed was that the power absorbed in sphere and emitted from sphere follows this pattern. It has a decay length which is similar to the decay length of unperturbed field, but the power scattered through glass and observable power, which is emitted at fluorescent stage, they have a shorter decay length, which means they decay faster with the bead depth. And this needs to be taken into account because we don't want to be in the situation where the bead goes out from the dynamic range of the microscope. The dynamic range should cover the entire length of DNA extensions so that we always get the visible, detectable photonic response from the bead. On the other hand, decreased decay length is good because it makes easier to detect small depth changes, since in this case the intensity change corresponding to the depth change will be higher. We studied this response for a variety of bead compositions and bead radius, and these results are similar in wide region, and we published these results. Over here there is a reference for a paper. It's published in Langmuir Journal of American Chemical Society. Now, it's same for larger beads. In our experiments, we are using beads of 4 micrometers in diameter. You can see here that on the picture below, you can see the isosurfaces of power absorbed inside the bead, and the pattern is similar. Inside the bead, there is 1,000 times less power absorbed than in the outer shells of the bead in both cross sections. So the question is, why not using this property and designing a bead where we have a magnetic core and fluorescent shell? This way, the magnetide will not interfere with the detectable visible signal from the fluorophore, and also no signal will be absorbed inside magnetide, and less energy will be transformed to heat. Thus, it will not increase the Brownian motion and not contribute to noise. We extended our numerical simulations to cover these processes. And on this figure, you can see our model of a bead with a magnetic core and fluorescent shell. And you can see that it works in simulations. In the magnetic core, the observed power is on a log scale around 1 million times smaller in the center and maybe 1,000 times smaller on the edge of the core. And the most power is observed here in the outer shell of the bead. And we're going to continue studying such properties of such beads to come up with a design which improves our sequencing sensitivity in future. And also, other direction we recently started to work in is the coatings of noble metals over both beads and substrates. There is a huge amount of literature onto the topic of surface plasmon resonances, and we're going to see how these effects can help us to achieve higher sensitivity of DNA sequencing. On the left, you can see the near field distribution in case of pure polystyrene bead with magnetite and fluorophore over a substrate. And on the right is the same bead, but with a coating of 50 nanometer thick gold layer, and the substrate has similar layer of gold as well. What you can see is that the field distribution is significantly changed. And we're going to study now how these changes are transmitted to actual observable power of the beads on the screen, and if we can manipulate the bead composition to achieve highest possible sensitivity and be able to detect sub-nanometer scale changes in DNA lengths. So to sum up, we're developing a fast and cheap whole human genome sequencing instrument. And so far, we designed the magnetic tweezers instrument to apply targeted high pulling forces to our chamber hosting DNA molecules. We constructed the actual device and are using it now in the prototype. Also, we designed the numerical code, which is capable of simulating optical response from magnetofluorescent beads. And we're going to study the fundamental properties of electromagnetic field scattering and absorption on micro and nanoscales to ensure that we can achieve high sensitivity of DNA elongation imaging. And I would like to thank Professor Fridon Shubutidze, who is supervising the research on the numerical part here in Thayer School of Engineering, and also our colleagues from Lehigh, Pearson Oliver and Professor Dmitry Vezanov, and Professor David Karkashadze from Tbilisi State University, who was visiting here last fall. And we had a lot of useful talks with him. And this project is funded by National Human Genome Research Institute. Thank you.
I think there's time for perhaps one or two quick questions. Chris?
Is spherical shape the optimal?
We haven't studied other shapes yet, but it's a good question. We can do that.
Another quick question, yeah?
In addition to shape, is there any advantage to a spatially or compositionally non-uniform proto-tissue interaction?
That's also a good question. Actually, Professor Gibson mentioned if we put a small thin layer of gold only on one part, creating like a cup, it may be beneficial. We can simulate it, but we have not yet. It's also part we're going to study.
Okay. If there are other questions, I'll ask you to hold them to the end of the second talk. Let's thank our speaker again. So our second speaker today is, I think this is working, John Lamppa, and he is a Ph.D. candidate at the Thayer School here and working with Carl Griswold. And starting in the fall of 2007, he's been working in the field of protein engineering with an emphasis on novel enzyme therapies for cystic fibrosis. He's going to tell you all about it. Cystic fibrosis and bacterial biofilm degradation. He received a BA and distinction in chemistry from St. Olaf College in 2007. Please welcome John.
Okay. So for the second part of the Jones seminar, I'd like to talk about what I've been doing here at Thayer for the last couple of years. And specifically, I'm going to be talking about enzyme therapies for bacterial biofilms. So before I dive into my specific project, I thought I'd just give everyone kind of a background on what protein engineering is, for those of you that might not have been even aware of the field. I don't know if there's a specific way to describe it at all, but the way I like to define it is a way to chemically or genetically modify a protein to increase its value for some specific function. And I just have a couple examples here that are currently available in the industry. And the first one is Aeruginosa, which we have over here. And if you saw George Georgiou's talk last Friday, he mentioned this enzyme a couple times. It's basically being used for leukemia patients. And the engineering that went into this enzyme was basically just to pegylate it, which is adding polyethylene glycol to the enzyme. And the reason why they did that for this enzyme was just to increase its size and basically increase the half-life in the body. And so that's one interesting enzyme. Another one that's getting quite a bit of use is Palmazyme, which is a recombinant human DNA. So basically, this enzyme right here cleaves DNA, so it cuts up the backbone of DNA. And that's being used for cystic fibrosis right now. Basically, in the lungs of CF patients, there's this really bad environment. There's lots of bacteria that's growing. And the body tries to attack that, basically sending an immune response such as neutrophils, and they can't basically get rid of this bacteria, so they lyse and they shed a lot of their extracellular DNA into the lungs. And so this enzyme goes through and kind of cleans it up for the patients and helps them cough up this kind of goopy mucus that forms due to that. And the last one I have up here is Puristar. This is one you might actually be using on a daily basis and not even know it. It was basically genetically engineered by Genencore to be more stable in very harsh conditions, such as those that use bleach. It's actually an enzyme that basically gets rid of starch, so stains in your clothes, you would use this enzyme to remove those. And so those are just a couple examples of what's out there currently. To give you an idea of the scale that we work on, this is alginate lyase. This is the enzyme I'll be talking about quite a bit throughout the seminar. As you can see, the diameter is only 8 nanometers, so it's quite small. And many of the mutations that we do on these enzymes to make them more stable or more active or whatever we're trying to do, I mean, we change basically just individual residues, so very, very small parts of an already small system. And so that'll kind of give you an idea of the level we're working on. So what kind of problems are we trying to address here at Thayer? Well, specifically, my lab is interested in bacterial biofilms. And on the left here, we have a river biofilm, which is not what we're interested in, but this kind of gives you an idea of what biofilms are like. So kind of that green, gunky, sludgy stuff that grows on rocks and debris in rivers. It's really goopy and thick. That kind of gives you an idea of what biofilms are like. And we're specifically interested in Pseudomonas aeruginosa biofilms. And in this picture on the right, we can actually see a kind of a red, pinkish microfiber here. And we have bacteria that's growing along it in green. And then we have kind of this brownish green biofilm that is actually helping support the growth of the bacteria. And it's secreted by the bacteria, and it actually helps it grow in more unfavorable conditions. And so this is what we're interested in, targeting these biofilms that basically don't do a lot of good things. They're known for corrosion of oil pipelines, dental plaque, helping infectious bacteria grow more readily. And so that's what we're looking at. And specifically, we're interested in biofilms that grow in the lungs of cystic fibrosis patients. And so those biofilms are secreted by Pseudomonas aeruginosa, and we're specifically interested in the mucoid phenotype. So it's a very robust biofilm that is produced by this type of Pseudomonas. And it's actually the leading cause of death in CF patients is this bacteria. And here I just have an X-ray picture. You can see some of this kind of white patchy material in each lung. That's actually scarring of the lung tissue caused by this very unfavorable environment that sets up in the lungs. And so basically this is kind of just a cross section of some biofilm here created by Pseudomonas. And that's, essentially what's going on inside their lungs. And so there are, antibiotics that do exist right now, but none of them are very good at actually penetrating this layer. And so this ends up being kind of the beginning of the end for CF patients when they contract this type of bacteria. So our solution was to, being protein engineers, was to go out and look for an enzyme or some kind of protein that possibly could get rid of these biofilms. And it turns out there's actually an enzyme known as alginate lyase. And this enzyme degrades alginate, which I have a structure of right here. And that is one of the main components of these mucoid biofilms. And so as of 2000, which was about 10 years ago, there were 72 known alginate lyases. So there are quite a few of these. However, they all have varying amounts of activity against different parts of the alginate structure. And what we did is we started going through these and trying to figure out which ones would work best for the alginate we work with. And specifically, Pseudomonas is composed mostly of mannuronic acid alginate. So you can have either guluronate or mannuronic acid as part of this structure. And so we kind of narrowed it down to a couple enzymes. And we ended up looking at one called alginate lyase A13, which I have pictured here. And so we had an enzyme that we thought would be fairly active, but unfortunately, when we were looking at all these enzymes, there are none that are actually human alginate lyases. So most of these are from a bacterial origin. And so that puts up a red flag right away. Like, you can't just give this to a patient and hope that nothing goes wrong. Most likely their immune system will notice that it's a foreign protein and will try to get rid of it. In a bad scenario, they could actually go into kind of a allergic shock to the protein and possibly even die from that being administered. So that was kind of the first hurdle we came up against, is this is the best enzyme we have to work with, but how can we give it to patients? And so we actually came up with this technique using pegylation, where we introduced various cysteine mutations throughout the enzyme, which I have labeled here. And we actually came up with a panel of these proteins where each one just had one of these mutations. And what this allowed us to do was to hook up polyethylene glycol, which is basically just a long, kind of carbon-like chain that can wrap itself around the protein and actually shield it from the body's immune system. And our main reason for looking at different sites of attachment is different parts of the protein can be recognized by the human immune system, and other parts maybe not so much. So it's good to look for a couple of areas, and you don't want to just put on too many PEG chains, because if they actually get within the active site here, they can cause the enzyme to be inactive, and so it wouldn't matter at that point if it had lowered immunogenicity if it couldn't actually function. And so kind of the big picture here is that this enzyme would be used as an inhaled therapeutic for CF patients, along with another inhaled antibiotic. And so the enzyme would chew through the biofilm, allowing the antibiotic better access to the infection. So kind of starting at the beginning of this project, we were talking to another group that actually worked with this enzyme, and we asked if we could have the bacterial genome so we could clone it out of the genome and start working with it. And they said no, they would not give it to us. So that was kind of interesting. So we actually sat down and we managed to synthesize it in the lab on our own. And we're able to come up with a fully functional folded protein, just like the bacteria itself does, that produces this protein. And kind of just to give you an idea of what the purification process is like to get this protein, we have a polyacrylamide gel here. And what this does is when you run proteins through it, large proteins run slowly, small proteins run through quickly. And so what we did is we got the DNA sequence for this protein. We put it into E. coli, which is a system that basically can make loads of protein for us. And what we have here in lane 2 is actually just native proteins from the E. coli. We then have E. coli that's expressing our protein, which you can see right here. And this band is not present in the native E. coli. And we then can go through a couple of purification schemes and actually get the protein on its own. At which point we then can add polyethylene glycol, purify that, get our pegylated product, and start testing activity to see, do we still have activity after we've pegylated this enzyme. And it turns out that this mutant here, where we pegylated this residue, had a maximal velocity that was just as good as the wild type, or possibly even better. So that was good. That was kind of the first thing to check off our list, is we had a pegylated enzyme that was still, fully functional. But this was all done against brown seaweed alginate, which is kind of just the standard for people that work with alginate lyases. But we were more interested in like alginate that's actually produced from a mucoid strain that's a clinical isolate of these CF patients. And what we did is we purified some alginate from FRD1, which is a clinical isolate strain. And we actually noticed that when we ran our best mutant here against the wild type, it was actually had a 1.8-fold increase in activity due to what we assume is the pegylation of the enzyme. And so that was kind of an unexpected result, but a good one for us. And so we said, okay, this is looking pretty good. So we moved forward and we decided to see, what is the immunogenicity that's involved with this enzyme. And the first thing we did is we gave basically the wild type enzyme to rabbits. And so rabbits, basically their immune system starts making antibodies against that. And you can harvest those out of the serum, and then actually see how well those antibodies bind to the various mutants you have. And we do that in the ELISA plate format. And what we saw is one of our mutants showed a 90% decrease in the amount of antibodies from rabbits that could bind to it compared to the wild type. And so that was promising, that looked good, but we were more interested in trying to get to a more therapeutically relevant scenario, which was looking at human antibodies. And what we did is we teamed up with some people at MIT, actually Margaret Ackerman, who'll be coming here next fall as a new professor. And they have a system where they actually have yeast cells that display human antibodies on the surface. And so what we did is we teamed up with them and we gave them our protein, and they basically mixed the protein with these yeasts that are displaying different human antibody fragments. And basically the amount of yeast that bind to our protein could then be plated, and you could count the number of colonies associated with that. And what we saw is our most active enzyme actually had a 91% decrease compared to the wild type in the amount of human antibodies that could bind to it. So everything was looking good at this point. So kind of the next step, one of the last things we could do in the lab before really moving into a more advanced living model was to grow actual biofilms, not just, before I'd been looking at soluble alginate, well, what about the whole biofilm structure that alginate is just a part of? And so what we did is we grew biofilms in 96-well plates, and I actually just have test tubes on here. But you can see the biofilm that's been stained in these tubes. And what we did is we basically just let the enzyme incubate with this robust biofilm. And what we saw was that both the wild type and our best mutant could strip the majority of the biofilm from the wells, with again the pegylated mutant being better than the wild type. And so this actually kind of shows real therapeutic activity, because this is the kind of biofilm that these enzymes would be up against, most likely in the lung environment. So what we're kind of working on now is actually trying to move into a mouse model to really see how well these enzymes work. And there are a couple of models out there currently, but we've devised a kind of a somewhat novel model that uses bioluminescent cells. So actually I have a vial of Pseudomonas here that's been engineered to emit light, much like fireflies do. And what we can do is actually have mice inhale this solution of Pseudomonas, and we can actually image the amount of light that is being given off from the bacteria inside their lungs. And at this point, we would then give them a treatment of both the alginate lyase and one of the antibiotics. And the idea is that if you can see a decrease in the luminescence, that means that the drug combination is working. And so we are just currently setting up this model at the DHMC. We haven't done a lot of work with it, but that's kind of where this whole project is headed at the moment. So with that, I'd just like to thank my advisor, Carl Griswold, and Tom Scanlon, who's been a very helpful postdoc on our lab, and also funding from the CF Foundation and the National Institute of Health.
So we have time for some questions.
Okay.
So the comparison between the human immune response to the wild type enzyme and then to one of your mutant enzymes that had a much lower comparable immune response. But I'm curious if that tells us anything about what the magnitude
Yeah, so I mean, really the only way to know that is just to give an animal the enzyme and just see, what is the size of the antibody production against that enzyme. And so that's the best way to do that, yeah. But they are two different things, looking at antibody binding and then also just what is the size of the response. So.
I have a question.
Yeah.
So the pegylated enzyme is doing a better job with the alginase. Do you have a model for why that might be? Is it a longer interaction time, perhaps?
Yeah, so we've actually been looking at that and trying to figure out maybe what the mechanism is behind that. And so far it's been hard to really set up a study where we can figure that out. I don't know. It could be that polyethylene glycol is quite good at solubilizing different objects. So maybe the alginate is more soluble when the PEG interacts with it. So
would that be a function of chain length? So that would help you track the mechanism at all?
yeah, so potentially a longer chain length could help increase the activity, or it could also get in the way of the enzyme.
Yeah.
So yeah.
Any other questions?
In your rabbit, in that slide right there, in your rabbit antibody response, you see much greater decrease for your S32 than the one that happened to be best activity with the A53. Was that just luck? That for rabbit, it wouldn't have been your best one, but for humans, you did even better? Or how would the S32 have done in humans?
Yeah, so we, when we teamed up with MIT, we only just sent them this enzyme, so they had so much time. But it would be really interesting to see if that one was potentially better. I don't know if it could get much better, but yeah, definitely. So.